Hello, welcome to this presentation. I am Kunjumon Vadakan. I study the nervous system to understand how it operates. I have a conflict of interest to declare. In the previous presentations, we have seen derivation of units of inner sensations by semblance hypothesis. In this presentation, we will see long-term potentiation into short-term LTP, an electrophysiological finding that has found correlations with behavioral markers of retrieved memories. So let us examine it. If we are associatively learning two stimuli, S1 and S2, using a mechanism that is, let us call it as mechanism X. So this is happening during associative learning. So mechanism X is induced by the associatively learned stimuli S1 and S2 at the locations of their convergence. And since we haven't seen any cellular change during memory retrieval, we can reasonably assume that there is a passive reactivation of the cellular change that is induced at the time of associative learning that takes place during memory retrieval, and memory is getting retrieved from this mechanism. If we are using a Q stimulus S1 at the time of memory retrieval, then that Q stimulus should passively reactivate the learning induced change X at the time of memory retrieval to induce units of virtual inner sensations of the sensory stimulus S2, and concurrent with that, we expect behavioral motor actions, because motor units are going to get activated and behavioral motor activity is going to get expressed either in the form of speech or motor action. So that is what is expected to happen at the time of memory retrieval. What we have observed is that the electrophysiological finding called long-term potentiation is correlated with the behavioral motor activities at the time of memory retrieval. So what we know is a correlation between long-term potentiation and behavioral motor activities at the time of memory retrieval. What we have to discover is the causation. So if we can hypothesize a mechanism X at the time of associative learning, whose passive reactivation is giving rise to units of inner sensations of memory concurrent with the behavioral motor actions, then from that mechanism we should be able to identify and explain how that mechanism X is giving rise to LTP. And if we can succeed in doing that, then that will help us to discover the mechanism that is operating at the time of associative learning and that is occurring at the time of memory retrieval. So that's the aim of this presentation. We have very large number of findings during LTP experiments that we can utilize. We can go and examine the hypothesized mechanism X and see whether that can explain all these findings of LTP. So let us go back to some historical events. In 1949, Professor Donald Hebb proposed that when an axon of a neuron is repeatedly stimulated, certain growth processes or biochemical changes are taking place at the synapses, and that is thought to be responsible for learning associated changes and changes that are occurring at the time of memory retrieval. Professor Hebb said that when an axon of a neuron, say for example if this is neuron N1, the axon of a neuron, this is presynaptic terminal, at the tip of that axonal terminal, and this is the presynaptic terminal, this is the postsynaptic terminal, or it's otherwise called spine or dendritic spine of the next neuron, that is N2. Professor Hebb postulated that when the axon of a neuron N1 is repeatedly stimulated, some type of growth processes or biochemical changes are taking place at the synapses. So those changes at the synapses were called as Hebbian synaptic plasticity changes. Certain other events took place since that period. In the middle 1950s, there was a patient called HM. Patient HM suffered from severe seizure disorder. Surgeons identified that the seizures are originating at the hippocampi of the patient HM. Surgeons removed both hippocampi. Following surgical removal of the hippocampi, seizures were stopped. But patient HM suffered severe memory problems. He could not learn anything new. Patient HM started forgetting whatever he learned in the previous 5 to 8 years period. However, his remote memories remained stable. This gave an idea that hippocampus is a location where memories are stored immediately after associative learning. So investigators focused on hippocampus, and it was reasonable to think that finding of any electrical change within the hippocampus that can last for a long period of time could be an indication of a cellular event that is responsible for memories. So this led investigators to put electrodes at various locations and stimulate and record from the output region to see whether they can discover any finding that can last for a long period of time. This led to the discovery of LTP. So let us see the experimental steps in LTP experiments. This is a neuronal order of CA3 neurons, and they have recurrent collateral axons moving parallel to each other. Another neuronal layer that is called CA1. So this is a CA1 neuron. They have their dendritic tree that cross across these axonal terminals. And these axonal terminals of CA3, they are called Schaffer collaterals. The dendritic tree, they have the dendritic spines. So we can draw 3 or 4 dendritic spines here. So these are the dendritic spines, and they synapse with the presynaptic terminals that are the axonal terminals of CA3 neurons. So these are the CA3, CA1 synapses. Now, what experimenters did was they put electrodes into this Schaffer collateral region. So this is an electrode. So this is a stimulating electrode, SE1. They stimulated this region, and then they started recording from this extracellular matrix space. So this is a recording electrode, and this recording from the extracellular matrix space is called field recording. This means that they are recording the electrical changes in this entire field. Initially, when they stimulated this Schaffer collateral area with a regular stimulus, they got a small regular response from this recording electrode. Later, when they stimulated this region with a high frequency stimulus, that's a high frequency stimulus. High frequency stimulus means a stimulus with high energy. So when they apply high energy here, they got a very high response. That was around 300% increase in the total potential. Following this high frequency stimulus, when they stimulated with a regular stimulus, then instead of getting a regular recording, they got a potentiated response that was nearly 300% again. So following the high frequency stimulus, a regular stimulus gave rise to a potentiated effect. The recording showed, and the response of the recording was like this. So there was an initial peak followed by a drop and a plateau. And they also noticed an initial delay of induction of this peak potentiation by around 30 seconds, and it is observed even up to 1 minute, and in some cases it lasts more than 1 minute. Another interesting observation was that these responses lasted for nearly 10 hours and even up to 24 hours. Now they have discovered something that showed a potentiation and that lasted for long period of time. A good model for explaining certain cellular changes that could be taking place at the time of associative learning. Later experiments showed that even when they put an electrode into this CA1 neuronal soma and when they record, and this type of recording is called patch clamping. Patch clamping recording of CA1 cell body also showed similar response pattern of this potentiated effect, a peak, a drop, followed by a plateau. Current experiments use mainly patch clamping recording because it's very specific to a neuron, and then therefore we can study the potentiated effect more specifically. Based on the synaptic plasticity hypothesis, the potentiated effect is occurring at these 4 synapses of the dendritic spines that belong to the CA1 neuron. It is a little bit surprising to see the 300% increase in the potentiation that has to arise from these 4 synapses only. In other words, the recorded CA1 neuron has fixed number of synapses. Can synaptic plasticity at these fixed number of synapses alone give rise to this potentiated effect? That is a question that needs to be answered. And the second question is why there is a delay of at least 30 seconds for the potentiated effect to appear. Can the hypothesized mechanism of synaptic plasticity thesis or that of the semblance hypothesis can explain the 30 seconds delay that is occurring at the time of LTP induction, in order to tell that LTP correlation with the behavioral motor activity is in fact indicative of a learning associative change, but why there is a delay? We have to find explanations. Investigators started inducing LTP by stimulating the dendritic spine directly to see what is the last step in the cellular process that is required to induce LTP. I am going to draw the synapse here, but it in an upside down fashion. So this is a presynaptic terminal of the CA3 neuron, and this is the postsynaptic terminal or the dendritic spine. These synapses are glutamatergic synapses, so glutamate is the neurotransmitter, and glutamate binds to 3 different type of receptors on this dendritic spine. Mainly they are NMDA receptors, AMPA receptors, and kainate receptors. Investigators blocked NMDA receptors on this dendritic spine, and then they directly stimulated these dendritic spines with voltage sensitive calcium channel activation to see whether they can induce LTP here so that they can record it from the CA1 neuron. Interestingly, they found that they could induce LTP by this method, and they got exactly the same shape of the graph. They got a peak, a drop, and a plateau after a delay of 30 seconds to 1 minute and even more than 1 minute. An exactly comparable potentiated effect was observed by stimulating these dendritic spines after blocking the NMDA receptors on these dendritic spines. So that indicates that the last step that is required to induce LTP at a synapse is the postsynaptic terminal. So we have to be very clear here. In normal conditions, synapses are required for the normal function. The stimuli have to come through the presynaptic terminal. There should be neurotransmitter release, and synapse have to activate it to activate this dendritic spine and the potentials to reach the neuronal soma. So this is an experimental paradigm to see the last minimum step at the synaptic level to induce long-term potentiation. So that indicates that the dendritic spines are the last step during the process of long-term potentiation. Now let us examine this observation in terms of the semblance hypothesis. According to Hebbian plasticity thesis, when an axon is repeatedly stimulated, there are some changes taking place at the synapses that is responsible for the changes that is likely happening at the time of learning or at the time of memory retrieval. But based on semblance hypothesis, when two stimuli S1 and S2 are activated at the same time, there could be a learning induced change at the locations of their convergence. So this is one synaptic pathway that is coming from one sensory stimulus S1, and there should be a second pathway that is coming from the second stimulus S2. So that is neuron N3, this is its presynaptic terminal, and this is its postsynaptic terminal, and this is neuron N4. So based on semblance hypothesis, certain changes should occur at the level between the postsynaptic terminals at the converging locations between these two stimulus, and its passive reactivation is responsible for retrieval of memory. At the time of memory retrieval, when stimulus S1 comes as a Q stimulus, it activates the synapse, depolarizes this dendritic spine, and it reactivates the interpostsynaptic functional link and activates the interlinked dendritic spine here. And at this time, two things are going to happen. Since this synapse is always getting quantum release of neurotransmitter release from the presynaptic terminal, depolarizing this postsynaptic terminal all the time, then any lateral activation coming from reactivation of this interpostsynaptic functional link is going to trick this dendritic spine to hallucinate that it received an activity from its sensory receptors that provides the unit of inner sensations of retrieved memories of the sensory stimulus S2. And the depolarization spreads through this dendritic spine, through the dendritic tree, and reach the neuron N4. And if neuron N4 is already at a subthreshold state, it is going to fire an action potential by the arrival of these additional potentials that is coming through this reactivation of this interpostsynaptic functional link, and it will trigger an action potential of neuron N4. If neuron N4 is connected to a motor unit, then this motor unit is going to get activated, giving rise to motor function. So now, based on semblance hypothesis, reactivation of interpostsynaptic function link cause both induction of units of inner sensation concurrent with the behavioral motor activity. Now, the question is, can interpostsynaptic functional link be explained here at these synaptic locations as a cause responsible for induction of LTP? Now let us see different types of interpostsynaptic functional links. Normally we expect there to be some water or hydration between the dendritic spines that electrically isolate these dendritic spines. There will be mild enlargement of these dendritic spines that will cause exclusion of that water or hydration. In other words, hydration exclusion happens so that these two dendritic spines come in contact with each other, so that the depolarization can spread from this dendritic spine to the next dendritic spine. Hydration exclusion requires very high energy. So high energy is required for that hydration exclusion. Because high energy is required, this process is highly reversible. It quickly reverses back. That is a good candidate mechanism for explaining working memory at the time of memory retrieval. And we have also seen that motivation promotes learning, and during motivation promoted learning it causes release of dopamine, and dopamine is a candidate mechanism for enlargement of dendritic spine. So anything that stimulates to enlarge the dendritic spines can then lead to the next step of the process, whereby we can get the hemifusion, both partial and complete hemifusion between the membranes of the dendritic spines here. So that is a membrane hemifusion process, and that is a highly reversible process. It can reverse back to single dendritic spines once the enlargement of the dendritic spine is getting reversed. We also saw that if the enlargement continues, this hemifusion gets converted to a step of fusion change. So now the membranes fused here. So that is the next advancing stage. From here to here, it is usually an irreversible stage. But if the size of the fused membrane is very small, it may reverse back. Now the question is, can we use these explanations to explain long-term potentiation and all its features? So for that, let us draw all the interpostsynaptic functional links that can be formed between the dendritic spines of different CA1 neurons at this location as responsible for inducing LTP. So let us draw few CA1 neurons and draw their dendritic tree and mark their dendritic spines, and let us see whether we can bring an explanation for formation of interpostsynaptic functional links as responsible for both learning associated change as well as the LTP. Now, based on the semblance hypothesis, very large number of interpostsynaptic functional links are formed between the dendritic spines that belong to different CA1 neurons. So how can we explain the potentiated effect? When high energy is induced at these Schaffer collaterals, the high energy leads to enlargement of the dendritic spines. Some of the dendritic spines are likely to be small within this field, and they are going to take time to enlarge, and they form interpostsynaptic functional links of different types. So for example, many of them will be of exclusion of water hydration, and many of them will enlarge further, will produce interpostsynaptic membrane hemifusion, and continuous enlargement will lead to interpostsynaptic membrane fusion. So these interpostsynaptic functional links will then interconnect all the spines, and the potentials will reach the dendritic spines, these 1, 2, 3, 4 dendritic spines that belonging to the neuron from which the recording is carried out. So that at a later time, when a regular stimulus is applied at the stimulating electrode, that stimulus will go through these axonal terminals, propagate through all these synapses, and then reach all these dendritic spines, and then they will summate and reach through these 4 dendritic spines to this CA1 neuronal soma from which recording is carried out. Since all these potentials get summated while arriving at the CA1 neuron, we can get continuous potentiated effect. So that can explain the potentiation. So that is one explanation by the semblance hypothesis. Number 2, the potentiated effect suddenly drops, and this drop is called short-term potentiation, or otherwise called STP. So how can we explain short-term potentiation? Sudden drop of the peaked potential to this plateaued level can be explained in terms of the quick reversal of the interpostsynaptic functional link that is formed by hydration exclusion. So since high energy is required, that gets reversed back very quickly, reversing many of these interpostsynaptic functional links. So now short-term potentiation can explain working memory. As the animal moves through an environment, it's going to associate many sensory stimuli. But many of these associations have to reverse back to independent dendritic spines so that animal can continue to associate many things as it moves through the environment. So short-term potentiation that is seen as the drop of the peaked potentiated effect can be explained in terms of reversal of wide hydration exclusion, and that's a good model for explaining working memory. And number 3 is the requirement for high energy to induce the LTP. High energy is required for the formation of large number of interpostsynaptic functional links because many of these spines have to enlarge to form interlinks between the spines so that the potentiated effect can arrive at the CA1 neuron. So that can explain the requirement for high energy. And now the question is how this potentiated effect can last for 10 hours and even up to 24 hours. Because we are using high energy, the dendritic spines continue to enlarge. The hemifusion is getting converted to fusion stage, and this is mostly an irreversible stage. Only very small fusion changes will get reversed, otherwise the big fused areas will remain as such, and that can explain why the potentiated effect can continue for 10 hours and even up to 24 hours. Now the most important finding here is a delay of up to 30 seconds and even up to 1 minute or even more than 1 minute, which is seen in almost all the LTP experiments, including those that were induced by activating the voltage sensitive calcium channels at the postsynaptic terminal region. So how do we explain this delay of up to 30 seconds? Associative learning takes just milliseconds for this change X to happen, whereas LTP takes around 30 seconds to get induced. So this is 1/1000 of a second, and here it is 30 seconds. So it is anywhere between 3000 to 30,000 times difference between the time scales of associative learning change and LTP induction time. So how can we explain that? Based on semblance hypothesis, LTP induction is taking place by the formation of large number of interpostsynaptic functional links, and many of them takes place by gradual expansion of these dendritic spines. Now the question is, do we have any evidence to suggest that interpostsynaptic functional links by the formation of this hydration exclusion, hemifusion and fusion, these stages can take time scales of seconds? In biological experiments, especially those that were carried out back in 1990s during the development of hybridoma cells, electric currents were used to fuse cells. So where large number of independent cells were exposed to fusion. Experiments that used both electric current and those that used no electric current, both these experiments have clearly shown time delays of 1 to even 10 minutes for fusion to take place between a group of cells. So fusion process is a slow process that takes time to complete. So therefore the continuation and the extension of this hydration exclusion towards the interpostsynaptic fusion process explains the time delay of 30 seconds to even more than 1 minute that takes place during LTP induction. Now investigators have found that after inducing LTP, the potentiated effect can be recorded not only from this CA1 neuron but also from adjacent CA1 neurons. If we put an electrode and record, we can find potentiated effect from neighboring CA1 neurons. This was thought to cause by non-Hebbian plasticity changes. Now we know that the interpostsynaptic functional link formation takes place in a large field here, and that causes interpostsynaptic functional link between dendritic spines of large number of CA1 neurons. So therefore, if we put electrodes in the neighboring neurons, we are going to get the same potentiated effect. So that can explain non-Hebbian plasticity. Another point is that motivation promotes learning, and during motivation it is known that dopamine is getting released. If this dendritic spine is receiving a dopaminergic input, we know that this dopamine release towards this dendritic spine cause the enlargement of this dendritic spine. So motivation promoted release of dopamine into the synapse that is formed with this dendritic spine can cause enlargement of this dendritic spine, and that can promote formation of different types of interpostsynaptic functional links. In the same way, we have known that dopamine can augment or increase the LTP induction. So that is another matching explanation between changes that takes place during associative learning and those that takes place during LTP. Another feature is that we told in the beginning that LTP is having correlations with behavioral motor activity. Now, since interpostsynaptic functional link formation can explain LTP, as we have seen in the beginning, the formation of interpostsynaptic functional links is responsible for learning induced change, and during memory retrieval what we are going to get are two things. One is the induction of units of inner sensations, and second one is the behavioral motor action. And now the formation of interpostsynaptic functional link can explain both LTP, and the same mechanism can explain the behavioral motor activity, and therefore we can explain the correlation that we have already observed between LTP and behavioral motor activity that is occurring through this reactivation of interpostsynaptic functional links. So at this point one would tend to ask the question, how can the interpostsynaptic functional link mediated mechanism takes place at physiological time scales during associative learning? We have seen that the high energy is required for the interpostsynaptic functional link formation by exclusion of water or hydration, and further enlargement is required to achieve these two stages during LTP induction. During associative learning, what we expect is that readily linkable spines undergo exclusion of water or hydration, as well as this interpostsynaptic membrane hemifusion that can take place at physiological time scales. So those are carried out by readily linkable dendritic spines. Now, as I mentioned before, even though interpostsynaptic functional link formation is responsible for both associative learning as well as LTP induction, one thing is very clear, that normal synaptic function is essential for induction of LTP, because the high energy has to propagate through the axonal terminals of CA3 recurrent collaterals and then arrive through these presynaptic terminals into the synapse. So normal synaptic function is required for induction of LTP. Similarly, here during associative learning, we need normal function of the synapse and normal function of the synapse to activate these two dendritic spines for the formation of the interpostsynaptic functional link there. And also during memory retrieval, the arrival of this Q stimulus activate this presynaptic terminal and the synapse is getting activated. So the normal synaptic function of this synapse is essential for depolarizing this dendritic spine and reactivating this interpostsynaptic functional link to reactivate this interlinked spine. So normal synaptic function is essential for the induction of LTP as well as associative learning process. So now let us see one of the glutamate receptors called AMPA receptors in the function of the interpostsynaptic functional link formation. So AMPA receptors are seen at the dendritic spine. They are located 25 nanometers away from the synaptic margin here. And we also know that One of the AMPA receptor subunits called GluR1 subunit is present within the dendritic spine inside membrane vesicles, and when they are activated, these vesicles move towards this postsynaptic location and exocytose their GluR1 subunit into this membrane region and are used to the formation of functional AMPA receptors. So, if this is the dendritic spine and if exocytosis takes place at this location, that excess of membrane segments of the vesicle is going to get incorporated at that dendritic spine membrane location, and that will lead to membrane reorganization at the location of these lateral segments that are 25 nanometers away from this synaptic margin. If you can draw another spine here, what we can see is that as a similar exocytosis can occur at the location of 25 nanometers away from the synaptic margin, and so that these membrane reorganizations taking place at these locations of interpostsynaptic regions can lead to the formation of interpostsynaptic hemifusion. So, that is a suiting candidate mechanism for the formation of both the exclusion of water hydration as well as interpostsynaptic membrane hemifusion, both partial and complete. So, again, during LTP induction, it takes time for these vesicles to move from here within this cytoplasmic locations to the postsynaptic margins, whereas during associative learning, it is the already docked AMPA receptor vesicles that are responsible for the reorganization of the membranes that can give rise to different types of interpostsynaptic functional links. Now, the beauty here is that the endocytosis of these AMPA receptor vesicles back to the cytoplasm is correlated with reversal of LTP. That means when the LTP is getting reversed back, the spine size reduces and the AMPA receptors are endocytosed into the cytoplasm. That means the membrane that is already reorganized here has to reverse back. That means some lipid membrane segments have to be taken from the postsynaptic membrane region and has to form vesicles, and those vesicles have to be transported to within the cytoplasmic region. So, the excess lipid membrane is lost there. So, that will naturally lead to the conversion of interpostsynaptic hemifusion back to the independent membrane state, and that can explain the reversal of LTP, and that observation provides further support for the interpostsynaptic functional link mechanism. Now, LTP experiments have shown that at near saturation stimulation conditions, that means at very high energy stimulation conditions, AMPA receptors are not required for LTP induction. What does it mean? It means that at very high stimulation conditions, the high energy that is delivered through the synapses towards the dendritic spines is sufficient enough to form the interpostsynaptic functional links, especially the last stage of interpostsynaptic fusion reaction. And no AMPA receptor is required. That means no need of vesicles to move towards the postsynaptic membrane region to reorganize the membrane and undergo hemifusion and fusion. High energy can cause the membrane enlargement, which can in turn lead to membrane hemifusion and fusion changes. The next finding is potentiation at single synapses. investigators studied the potentiation of ampere currents at single synapses. So investigators studied by stimulating the dendritic spine of a neuron. This is a neuron, this is its dendritic spine, and this is the presynaptic terminal. Investigators uncaged glutamate into a single dendritic spine, and then they recorded from this neuronal soma for the ampere currents. So when single dendritic spines were stimulated, they could record potentiation of ampere currents when recorded from its neuronal soma. How can we explain that? They found the potentiation at small dendritic spines, and those dendritic spines enlarged. Those dendritic spines show enlargement, and this enlargement was responsible for the potentiated effect. So it can be explained in terms of formation of interpostsynaptic functional links with the dendritic spines that are abutted. So they found the potentiation of ampere currents only in small dendritic spines that got enlarged. There was a finding that certain synapses do not have ampere receptors, since only ampere receptors can conduct currents at resting membrane potentials, and since these synapses were not having ampere receptors on the dendritic spines, those synapses were called silent synapses. So continuous stimulation produced appearance of the ampere receptors over the dendritic spines from the vesicles within the postsynaptic terminals, and that eventually made those synapses active. So that can explain the finding of silent synapses and their functional significance. Now we will see certain proteins within the postsynaptic terminal or within the dendritic spines that are important for both physiological functions of the formation of different interpostsynaptic functional links, as well as for the induction of LTP. And such a protein is called snare protein. So this is a presynaptic terminal, this is a postsynaptic terminal. In fact, they were initially discovered at the presynaptic terminals. Their functional role is already known. They are important for fusion of the synaptic vesicles with the presynaptic terminal membrane and exocytosis of the synaptic vesicle contents. Some types of snare proteins are also present at the postsynaptic terminal. Experiments using artificial double-layered membrane vesicles shows that the snare proteins can bring two membrane layers together. This is by both formation of an alpha helical bundles that forces two membranes together. It is also by self-organization of the snare proteins on these membranes. By both these methods, it can pull two membranes together and make them to undergo fusion process. Using artificial membranes, it was also found that both hemifusion and fusion are definitive end products of their action. However, at the postsynaptic membrane level, it is reasonable to expect that snare proteins can bring these membranes together to exclude the water hydration, as well as to make them to undergo a postsynaptic membrane hemifusion changes. However, we expect excellent checkpoint mechanism that prevent the formation of interpostsynaptic fusion change. It is also known that snare proteins are regulated by other proteins such as complexin, and together it is reasonable to expect that these proteins regulate the interpostsynaptic membrane hemifusion stage and will prevent any further conversion of that hemifusion stage to fusion stage. And also blockers of snare proteins, when it was injected to the cell body of this postsynaptic neuron, that blocked LTP. So that clearly indicates that blockers of snare proteins into the cell bodies of these postsynaptic neurons prevented snare mediated fusion changes between the dendritic spines of the abutted spines, and that reduced the LTP. So that's a clear indication that the snare proteins are responsible for fusion changes between the spines at the time of LTP induction. Now we will see some of the features of long-term potentiation that were explained previously. One is called cooperativity. Cooperativity means a threshold stimulus intensity is required to stimulate this stimulating electrode to induce long-term potentiation. That means that the stimulus has to activate a critical number of these axonal terminals to stimulate a critical number of these dendritic spines between a set of CA1 neurons to form interpostsynaptic functional links, so that at a later time a regular stimulus will go through those connections and the potentials will traverse and propagate through the interpostsynaptic functional links, get summated together to arrive at this recording CA1 neuron to give rise to the potentiated effect. So that's an explanation for cooperativity. Now we can explain associativity and input specificity. To do that, I have drawn an additional stimulating electrode SE2 here that stimulates a group of axonal terminals of the additional CA3 neurons that resulted in the formation of a small group of interlinked spines here. So a regular stimulus at this stimulating electrode SE2 is expected to form interlinks between spines that belong to different CA1 neurons at this location, and that interlinked dendritic spines has no connection with the large group of spines that are interlinked with the high frequency stimulation. So when we stimulate with a regular stimulus this SE2, that will not result in any potentiated effect when recorded from this CA1 neuron. However, when a high frequency stimulus is applied to this SE1 and a regular stimulus is applied at this SE2 simultaneously, that will result in the formation of an interlink or interlinks between two spines or more number of spines between this group of interlinked spines and this large group of interlinked spine, so that at a later time when a regular stimulus is applied at this SE2, it results in a potentiated effect when recorded from this CA1 neuron. So what is going on here is that an interpostsynaptic functional link is formed between this spine that belong to this group and this dendritic spine that belong to this large group, so that when stimulated by a regular stimulus, the potentials that are generated within these dendritic spines will propagate through these interlinked postsynaptic terminals, and the potentials will summate and reach at this CA1 neuron that is recorded from this recording electrode as a potentiated result. That explains associativity. Now what does input specificity mean? Now I have also drawn a stimulating electrode SE3 here, whose stimulation with a regular stimulus has resulted in the formation of a small group of interlinked dendritic spines. When a regular stimulus is applied to this stimulating electrode SE3 together with a high frequency stimulus that is applied with this stimulating electrode SE1, then an inter-postsynaptic functional link is formed at this level between this small group of dendritic spines here and this large group of dendritic spines, so that a regular stimulus applied later at this stimulating electrode SE3 will result in a potentiated effect when recorded from this recording electrode. Input specificity means the interlink between groups of dendritic spines depend on which groups were stimulated together. For example, if this high frequency stimulus, if it is paired with this regular stimulus, then interpostsynaptic functional link will form only at this location. Whereas if the high frequency stimulus at this stimulating electrode is paired with a regular stimulus at this stimulating electrode, then an interpostsynaptic functional link is formed at this location, interlinking this group of interlinked spines and this group of interlinked spines, so that at a later time stimulation with a regular stimulus at this location or this location will correspondingly produce a potentiated effect when recorded from this CA1 neuron. So that explains input specificity. Studies have shown that in order for input specificity to explain, it is required that the distance between the stimulating electrodes should be at least 70 micrometers away from each other. That means this group of interlinked spines should be separated from this group. Otherwise, they will form part of a single group. Now, the next feature is occlusion. Occlusion means that the formation of interpostsynaptic functional link that takes place during associative learning can be occluded with formation of interpostsynaptic functional link that is formed during LTP induction. Say for example, let us imagine that we induce LTP using this high frequency stimulus, and so that a group of interlinked spines are formed here. And if we then try to use these two dendritic spines for the formation of an interpostsynaptic functional link during a specific associative learning event, that will not result in the associative learning. This is because during memory retrieval, when the cue stimulus arrives, it is expected to reactivate only this interpostsynaptic functional link in order to induce units of inner sensations for a specific memory of this S2. Since this high frequency stimulus has induced interpostsynaptic functional links between large number of dendritic spines, the cue stimulus that arrives at this dendritic spine, instead of getting propagated to the interlinked spine alone, it is going to get propagated to a large group of dendritic spines here as a result of the formation of a large group of non-specific interpostsynaptic functional links, and that will induce semblances at all these interlinked spines, so that it will dilute the net memory that is formed. So there won't be any specific memory retrieval. So this indicates that a prior LTP induction at a specific location will occlude later learning. And the converse is also found to be true. That means if you use two spines to associatively learn, that will produce an interpostsynaptic functional link. And if you try to induce LTP between these two dendritic spines that already took part in the associative learning, then we will not be able to induce LTP between these two dendritic spines. Experiments have tried to induce LTP locally between specific dendritic spines. So once interpostsynaptic functional link is formed at the time of associative learning, then that will raise the baseline, so that when we try to induce LTP between these two dendritic spines, it will not show any LTP because the baseline has already got elevated because of the formation of interpostsynaptic functional link as a result of associative learning. So that can explain how prior learning can occlude later LTP between two specific dendritic spines. Now let us examine another phenomenon that is called kindling. The minimum amount of high frequency stimulus that is required to induce LTP is already known, but if you stimulate with higher frequencies of energies at this location, then instead of inducing LTP, it is going to induce after discharges, and in live animals that lead to a phenomenon called kindling, and the manifestation of kindling is seizures. In a separate presentation, I will be presenting a mechanistic explanation for seizures. Seizures are explained by the generation of large number of interpostsynaptic functional links that can propagate from one area of the cortex to other remote areas of the cortex. So if you use high frequency stimulus with much more energy than it is sufficient for inducing LTP, then it can lead to the generation of very large number of interpostsynaptic functional links between very large number of dendritic spines, and that can lead to the phenomenon of kindling. And it is of interest to note that the mechanism for associative learning involves formation of different types of interpostsynaptic functional links, and it is at those locations that we can induce robust LTP and kindling. Say for example, hippocampus. Hippocampus is a region involved in associative learning, and also is a region where we can induce robust LTP and kindling. So this indicates that a spectrum of changes ranging from hydration exclusion to formation of interpostsynaptic membrane fusion can explain how this range of changes occurring during associated learning to LTP to kindling can take place. So that is a demonstration that how the spectrum of changes can explain the spectrum of phenomena. Now we will see another feature at one of the regions called CA2 region in the hippocampus. At CA2 region, it is highly resistant to induce LTP. We have seen that at this CA3, CA1 synaptic region, we can induce LTP, whereas at the CA2 region, we cannot induce LTP. And investigators have found that the neuronal processes at that region is covered by perineural net proteins. So for example, if this is a synapse and if this is another synapse, these synaptic processes are covered by perineural net proteins. So these proteins can prevent LTP based on semblance hypothesis, because it prevents the formation of an interpostsynaptic functional link that is essential for the induction of LTP. Investigators have also discovered a method to dissolve these perineural net proteins, following which LTP can be induced at the CA2 region. So that provides an explanation for the formation of interpostsynaptic functional links during LTP. And interestingly, CA2 region is highly resistant to seizures. We have just seen that seizures can be explained in terms of kindling. That means very high frequency stimulus can cause a rapid spread of depolarization through the dendritic spines by the formation of interpostsynaptic functional links. And because of the presence of the perineural net protein, it resists any such spread, and CA2 region is found to be highly resistant to seizures. So that provides additional explanations why CA2 region is resistant to seizures. Another finding associated with LTP is that LTP induction by a high frequency stimulus alters place field. So it alters place fields. What does it mean? Place cells are CA1 neurons. When the animal is at a particular place in the environment, it receives Q stimuli, and the Q stimuli, as it arrives, it reactivates the interpostsynaptic functional link and induce inner sensation of the memories of its location, and it also the depolarization also spread through this interlinked spine through the neuron N4, and if this neuron N4 is at a subthreshold level, it is going to fire an action potential, triggering a motor movement. So this neuron is going to trigger an action potential when the Q stimulus arrives. So in a similar way, the place cells specific to a place or a location in the environment can receive sensory stimuli if those CA1 neurons are at a subthreshold level at that time. Then the additional potentials that are arriving through these interlinked spines can lead to the firing of specific set of CA1 neurons at a specific place or a location. If we induce LTP, that is going to result in the formation of interpostsynaptic functional links between dendritic spines of a large area. So instead of firing of specific set of CA1 neurons that are specific to a specific location, a group of CA1 neuron is going to fire non-specifically. That explains why place fields are altered after the induction of LTP. Another feature is that instead of this high frequency stimulus, we can provide this high energy by different methods. For example, a single burst with a high energy can induce LTP. So one of the requirements is that such high energy single bursts require dendritic spikes in order to induce LTP. So let us see what that means. Say, for example, if this is the neuron and this is the neuronal soma, this is the dendritic tree, it receives inputs through large number of dendritic spines. So for example, I am drawing just 2 dendritic spines, and this is the axon and these are the axonal terminals. When this neuronal soma receives inputs of nearly 40 to 100, these postsynaptic potentials will summate at this axonal hillock and give rise to a somatic spike. In other words, it's an action potential. So similar spikes are also observed at this dendritic region. So for example, it is observed that there are similar spikes at these dendritic regions. These dendritic spikes are the results of summation of 10 to 50 postsynaptic potentials. We still don't know how these postsynaptic potentials are getting summated to induce dendritic spikes, but these dendritic spikes are comparable to the somatic spikes that are induced at this axon hillock region. In order to induce LTP by a strong stimulation by a single burst, it was known that dendritic spikes are required. So that means large number of postsynaptic potentials have to be summated at those dendritic regions in order to induce LTP. Dendritic spike is a summated potentials that are arriving from a group of dendritic spines at this region. And we have explained the induction of LTP in terms of the formation of interpostsynaptic functional links between a large group of dendritic spines. And based on the semblance hypothesis and based on this mechanism, the single burst of strong stimulus can induce dendritic spikes by the formation of interpostsynaptic functional links between large number of dendritic spines. And it is known that at this dendritic tree, the dendritic spines are located at certain distances. On an average, the average spine diameter is less than the inter-spine distance, that is on an average. So that shows that the dendritic spines between which the interposed synaptic functional links are formed should belong to different neurons. So based on semblance hypothesis, the dendritic spikes are the results of summation of postsynaptic potentials that are induced at dendritic spines that are interlinked and that belong to different neurons. So this provides an additional explanation for the induction of LTP by the strong single stimulus that requires dendritic spikes. To summarize, we have explained large number of features of LTP in terms of the formation of interpostsynaptic functional link formation. What we were knowing so far was the correlation between LTP and behavioral motor activity. And now we have an explanation for the causation of both LTP induction and associative learning. Particularly, we were successful in explaining the delay of 30 seconds to 1 minute for LTP induction using the same mechanism that takes place at the time of associative learning. We have specifically noted that interpostsynaptic functional link formation at the time of associative learning takes place between readily linkable dendritic spines, whereas after the induction of LTP, the small spines require time for their expansion so that interpostsynaptic functional links can form between large number of dendritic spines so that a regular stimulus can propagate through these interlinked spines and the potentials can get summated so that they can be recorded at this CA1 neuronal soma as potentiated effect. So I sincerely hope that you understood these explanations, and if you have any questions, please feel free to email me or ask me below this video. I will be happy to answer them as much as I can. Thank you and have a great day.